Oliver Niels is now a radiopharmaceutical chemist at the German Cancer Research Center in Heidelberg, Germany. He is head of production and qualified person responsible for PET radiopharmaceuticals. His research interest covers the translation of novel PET tracers from preclinical research into the clinic. And he is also a radiopharmaceutical coordinator of a prospective multicenter clinical trial within the German Cancer Consortium using gallium PSMA 11.
Thank you very much.
The word is yours.
Professor Filligan for the kind introduction. One correction, I'm just a co-coordinator. The main coordinator is Klaus Kopka, he's sitting in the audience as well. I would like to introduce, it's the first slide of Melbourne. I've been a postdoc in Melbourne from 2007 to 2011, so we are sitting down in the left bottom corner. That's where the Melbourne Exhibition Centre is. Some of you have been to the new Victorian Comprehensive Cancer Centre, which is a little bit more north located in Melbourne distances. That's just a short walk. And I did my work at the old Peter Mac location in East Melbourne. Together with these three guys. So on the left, that's Peter Roselt. He's a radiochemist, and that was my major colleague over there. In the middle, it's a quite well-known nuclear medicine specialist. On the right, it's Peter Yu. He's a radiopharmacist, and neuroendocrine tumor therapies have been done in Melbourne for a couple of years. And there was not really a diagnostic tool to stage the therapy. And in 2008, we first got a gallium-68 generator initiated by Peter Yu on the right. And we got it in 2008, but we had no apparatus to do synthesis. And that's when the chairman, from your view on the left, suddenly stepped up in our lab and installed this apparatus on the right, which is a very easy apparatus to label gallium peptides. So it's a manual apparatus, and I'm not sure how many chemists are in the audience, but it's very easy. There's no. Okay. In the middle, there's the generator that's eluted with hydrochloric acid. Then there's a pure purification, and the radionuclide ends up in the middle in the reactor where the peptide is already located. And after labeling, the peptide is taken out on the left on a C18 cartridge where it's purified, and then it goes into the left for sterile filtration. From training, I'm a carbon-11 and fluorine-18 chemist, so I was really thrown into cold water when I got the task to do this job. I had to get familiar with gallium-68 labeling, and for quality control, what I did is I need to do an HPLC. Some chemists, they only did TLC and cut it in half and measured the activity. And I was thinking, okay, we need to do HPLC to be really sure that it's pure. Surprisingly, what we got in the first results was an HPLC chromatogram, which you can see on the left. That's a quite pure gallium dotatate chromatogram. The only reason it looked like this because I was lucky enough to get a vial for a lutate preparation where all the ingredients were listed on the vial, basically gentisic acid and ascorbic acid. The importance of these compounds is that they work as scavengers for radiolysis, which has been shown by the Erasmus group from Rotterdam. So if you only use water to dissolve your peptide, you get a chromatogram like on the left bottom, the control. If you add some ascorbic acid, it looks a bit better. If you add ethanol, still looks not too bad. Some gentisic acid, also okay, but the whole mixture of gentisic acid, ascorbic acid, and ethanol make the chromatogram look like this. So you really have a 95 plus pure radio-labeled peptide. And if you want to use higher activities of gallium-68, it's also very important that you don't occur radiolysis, and that has been shown by the group from Roger Schibli and PSI. Going back to the synthesis unit, one of the few drawbacks this unit has is it was a purification with acetone and hydrochloric acid. And if we use this solution to purify it, suddenly over a couple of weeks and months, an unknown impurity appeared in our HPLC chromatograms, and we didn't really know what it was. And I've been in touch with the Innsbruck group, Clemens de Cristoforo, and he published it a couple of weeks later that the hydrochloric acid and the acetone, they form a condensation product, which is then being shown up in your HPLC chromatogram, and that can be the problem can be dissolved if you just store it at -20 in the fridge and make aliquots and take them fresh out of the fridge. My conclusion on this HPLC or quality control problems is, because this is basic research more or less, that was 2008, there was no monograph on any gallium compounds, but you still need scientists to find out these things, and I would like to encourage the scientists who are in the audience to publish these things, because from practical aspects, if I stand in the lab and don't know these problems, it takes a lot of time and brainstorming to get better results. So when my time was over in Australia, I moved back to Germany, to Heidelberg, where I'm now, and I had to get familiar with the legislation in Germany, which is somewhat different to Australia. So we have a section 13.2b of the German Pharmaceutical Acts, which is mainly used for new compounds, in particular for gallium-labeled peptides. And it says a manufacturing license is not required if a medical doctor is responsible for the application of a pharmaceutical for one individual patient. So we're talking about one dose, not making a batch, and you can use it for 4 patients or 3 patients, just for one. And the responsibility includes also not only the application, using the syringe inject, but also state-of-the-art manufacturing of the pharmaceutical, which includes batch production and quality control. But this can be delegated from the physician to the chemists. The second option to produce radiotracers is obtaining a manufacturing license, which allows in-house production for multiple patients. And this manufacturing license is required if you want to conduct a clinical trial with an IMP. And the highest level is obtaining a marketing license, which also allows you to distribute your tracer. And for PET tracers, the only one in Germany is currently FDG. As I'm now in Heidelberg, it's unavoidable to talk about PSMA. The net tracers are still used, but our main topic is obviously PSMA. And the first PSMA ligands have been established about 2001 from the US groups from Alan Korzakowski and Martin Pomper and John Babbage. The first clinical application was conducted in 2011 in Heidelberg, but still it took a bit of time to get it into the clinic and see published clinical results, obviously in 2015, where we had 17 publications on PSMA PET ligands. This is an overview of the most used PSMA ligands based on the urea motif. The most used one is PSMA 11 from our group. Then the theranostic variant is PSMA 617. The Munich group has developed a PSMA INT ligand, and especially for radiochemists and radiopharmacists, the production of gallium tracers is very work intensive, because the PSMA ligands are requested very, very frequently from the clinic. So it's very important to get a F18 labeled PSMA ligand in the future, and 3 of them have already been published and also brought into the clinic. That's the 2 Pomper compounds, 18F TCFPC, TCFPYL, and the most recent one from our group in Heidelberg, PSMA 1007, which is a theranostic variant, or you can use it as a tandem with PSMA 617. To go back on the PSMA 11, the first preclinical results have been published by Matthias Eder under supervision of Michael Eisenhut in 2012. And 1 year later, Ali Afshar-Ormi published a clinical paper with 37 patients showing the superiority against fluorocholine. And because of the rising number of patients, it's very necessary to translate this tracer into the clinics under GMP compliance. And the managing board of the German Cancer Research Center came up in 2013 with the idea to conduct a multicenter clinical trial with 11 centers. And he supported us to do that. The aim of the trial is to show the safety and diagnostic accuracy in newly diagnosed prostate cancer in high-risk patients, and to image prostate cancer tissue within the prostate glands and pelvic lymph node metastasis. One main prerequisite to do this multicenter clinical trial is a functional radiopharmacy. The requirements are a radionuclide generator, a GMP-compliant production, and a quality control corresponding to the European Pharmacopoeia, including GMP documentation, which means a lot of so-called standard operating procedures, SOPs. For all centers, it's a requirement to apply for a manufacturing authorization, and they need to be carefully harmonized with all participating consortium partners. And in this study, we have 11 partners, and the difficulty is also that it's not a centralized production, but a decentralized production. The tracer is manufactured at 11 different sites. We have guidelines in the European Union which describe what is required to conduct a multicenter clinical trial under GMP guidelines. So it's a list of things we have to be careful, and all 11 sites need to fulfill these requirements: having a clean room, appropriate premises, personal equipment, a QA system, documentation, production, quality control. And there's another annex in the European GMP guidelines, the Annex 13 for IMPs. And I give you later a little example what that holds in. We're now in the year 2013, where we've been asked to do this clinical trial. Monographs have been established for gallium chloride solution for radiolabeling and for gallium dotatate. These are binding, but validated methods and procedures are possible. What is the future? The future for European monographs is that there's a monograph in elaboration for gallium PSMA and also for DOTA-NOC and dotatate. So when we started this multicenter clinical trial, we need to have a look what is available in all different centers. We have 11 centers, but every center has used a different generator. And every center has used a different synthesis module. And as I said before, all these conditions need to be harmonized. Ideally, everyone uses the same generator, the same synthesis module, but that will cost a lot of money to do that. Starting with the radionuclide generator, that was the first pitfall of our study. When we started, no germanium-68, gallium-68 generator with a marketing authorization was available until 2014. And that was a big remark from inspectors. The generators labeled with the sticker "not for human use" is not acceptable. So what did we do? We validated all the generators used in the centers according to the European monograph gallium chloride solution for radiolabeling, which includes testing for radiochemical quality, chemical quality, biological quality. After 5 to 6 months, we evaluated all generators. And then Eckert and Ziegler obtained a marketing authorization for their generator, and this one became mandatory for the clinical trial. The second pitfall we had in this preparation of a clinical trial, that we need to harmonize the synthesis procedure. So we need to do an identical manufacturing process. Every site has to do the tracer the same way. We decided to go for radionuclide pre-purification using a cation exchange system and harmonize the labeling conditions. The amount of precursor we use was limited to available tox data and suitability of labeling. We had the option to choose between two different types of buffers. One is acetate, the other one is HEPAs. And due to the non-reproducibility of the test for HEPAs, we decided to go for acetate buffer. The HEPAs test is described in the European monograph of gallium dotatate. Then the duration and temperature had to be the same. We went for 10 minutes, 100 degrees. And then the product purification and formulation has to be identical. So each batch has to have the same amount of ethanol. We need to make a choice between which sort of buffer we use. One synthesis kit came with sodium chloride solution as formulated product. A kit from another synthesis module came with PBS buffer. So we had to decide, okay, which one we use. Then the final product had to be defined. We're now doing a final product volume of 16 milliliters, and that's also based on the toxicity data. And most important, availability of GMP-compliant disposable kits for different types of synthesis modules. If you have a synthesis module, it does not mean that your kit is GMP-compliant. And it also has to be the same, it has to do the same radiosynthesis. Quality control, we took orientation on the monograph of gallium dotatate, set all the parameters. The limitation was the content of PSMA 11. Based on the tox status, it's limited to 0.6 micrograms per mil of PSMA 11. If you would use more PSMA 11, your yields would obviously go up. That's where we are limited. Quality control of gallium PSMA is also an issue. It's been shown here. I got a lot of calls and emails about your labeling of PSMA works really fine, but we always get 2 peaks. And we did some investigational research afterwards, because it's obviously published very early in the last century from Le Platinier in 1967, and then 30 years or 25 years later from a scientist at the German Cancer Research Center, that we have diastereomers being formed due to the nature of the chelator. And the reason why you get 2 peaks is that it's depending on the room, or on the temperature of the labeling. So if you label at room temperature, your right peak gets larger. If you heat it up, your left peak gets larger. But the cell uptake and the binding affinity for both mixtures remains the same. So 2 years later, we're at a stage where all the centers are using the same generator, and the type of radiosynthesizers has been brought down to 2. We had a couple of inspections so far from different centers. The difficulty is that we are working in 3 different countries, and they all have 3 different legislations. So it's in Germany, Switzerland, and Austria. Germany, in addition, has also 5 federal states which need to follow the German Pharmaceutical Act, but each inspector has a different interpretation of how to use the Pharmaceutical Act. I would give you some examples, which comments we got. Incoming goods control. How do you make really clear that your PSMA is your PSMA, if it's nothing else? And if you use 10 micrograms per synthesis, it's very hard to do a good incoming good control with analytical techniques. We should do a suitability test of disposable kits. We need to make sure that if we run a synthesis, that nothing that sits in this kit eludes out of the kit into the product. And it's very important that sufficient documentation is provided for the generator, the precursor, the disposable kits, and, as I said before, a sticker not for human use or for research purposes only is unacceptable, which makes it a bit difficult because we are using a complete new radio tracer, which is not produced under GMP for a couple of years. Most important part is without IMPD, that saying is derived from a radiochemist saying no tracer, no fun. I just translated into no IMPD, no fun. If you don't have an IMPD, you will not start any clinical trial. And this is how an IMPD is composed. So there are a lot of chapters you need to follow, you need to write down, a lot of documentation. And that's the pitfall number 3. How do we get an IMPD if we have 11 centers with so many different variations of generators and synthesis modules? The IMPD needs to contain information about the precursor, the process of synthesis, and the quality control of the IMP. There are a couple of templates and recommendations for radiopharmaceuticals available. In particular, from the ENM, they have written some guidelines. And our decision was can we harmonize different prerequisites for decentralized productions. We set the minimum requirements in our IMPD, and now it's fingers crossed that that we get green light from our authorities. This is an example how we need to follow the guidelines for Annex 13 for IMPs. That's a primary packaging sticker. And the annex says what has to be on the sticker. So it's name, address, and phone number of sponsor, CRO, investigator, pharmaceutical dosage form, the batch number to identify the contents and packaging operations. Very important, a trial reference code, trial subject ID number, that's the patient code, directions for use, how to apply the tracer, a wording for clinical trial use only, or something similar. Storage conditions, which is for PET radio tracers a bit, you can put it on, but usually it's used within a couple of hours, and that's what we said as well. The period of use in a manner that avoids any ambiguity, and most important, keep it out of reach of kids. But that only applies when the product is being taken home. So these stickers have to be put on, I don't know how many chemists or physicians are in here, it has to be put on one of these little vials. So it's very hard if you have so much information. So the primary packaging sticker goes onto that vial, and it's very hard to make it clearly visible. That's the option as well you need to fulfill. So to sum up what we've done, a couple of centers have already applied for manufacturing authorization. We are still in the pre-final stage of composing an IMPD. A CRO to organize and accompany the study has been finished in 2016 in May after EU government procurement, so we couldn't even hire a CRO, we just need to do a procurement within the European Union. The study protocol has been finished in August 2016, and protocols for Ethics Committee, BFS, that's the Radiation Protection Agency in Germany, and the national authorities will be submitted very soon. So the ethics submission has been done, BFS has been done, and we now will submit to BfArM, Swissmedic, and AGIS, which is the authority in Austria and Switzerland. The German Cancer Center, German Cancer Research Center, has been determined a sponsor in 2015. We got a DKTK, which is a German cancer consortium. We got approval for a joint funding project since January 2016. It's about 500,000 euros, which are then spread on all sites. Urologists and pathologists are now being involved in all participating sites. Clinical pre-study visits in the nuclear medicine departments are almost finished. I think there are only 2 or 3 left. And the study visits of all radiopharmacies have started in October 2016. So we visited the first site, and 10 have to be done. And to conclude, I would like to say that communication within such a multi-center clinical trial is very important. The communication between the participating sites, but also with local and federal authorities, is very important, and it's a key to get this clinical trial up and running. So we started in 2013, and the first patient hasn't even been done. It's a lot of work. And I would like to thank you for your attention.
Thank you very much, Oliver, for a very thorough talk and very valuable details. And this presentation is open for the discussion. Any questions? Yes, please.
David Dick, University of Iowa. Very informative talk. Could you comment on if, for the trial, you have any minimum or maximum times from the previous elution? So, like, do you have to elute the generator between 2 and 8 hours before the synthesis, something like that? And then also comment on what your specifications are for determining breakthrough.
I can comment on the last one. The specification for germanium breakthrough is taken out from the European monograph. I think it's, from my memory, 0.001%. That's a specification from the European pharmacy.
Are you requiring that on every run or periodically?
We test it periodically, once a week. But as we use now a licensed generator, for the national authorities, it's okay to use it. So we don't even have to determine the germanium breakthrough. It's a recommendation which is given by the supplier of the generator. So if you use the licensed generator for our federal authorities, it's okay to use it as it is. So we don't have to do the tests from the monograph in the European Pharmacopoeia.
And then the first question, do you have any specification for when you have to have eluted the generator before the synthesis?
That's what we do it how it's described in the manual for the generator. From my experience also here in Melbourne, you should use it maybe, or you should elute your generator at least 24 hours before use, but ideally if it's eluted 4 hours before use, that's sufficient. But maybe we have an expert
that's
sitting
the
here
recommendation. There is no specification in Pharmacopoeia telling about that, but the general recommendation is to pre-elute it before the synthesis, because the accumulation of stable zinc, which is the product of gallium decay, and other metal cations tend to accumulate in the generator. So the shorter the time before the pre-elution, the better synthesis you will have. But it will also depend on the amount of your ligand or your precursor. So sometimes it might not even make any difference because you have so large excess that compensates for the presence of metal cation impurities.
Maybe I can add another comment. We also had difficulties. It's the typical Monday morning elution when the generator was sitting still for the whole weekend and you had to do a synthesis Monday morning, 7 or 8 a.m.
Yeah.
And the generator wasn't eluted for the whole weekend. Then you really have problems in your synthesis. So what we did is we made an automated module where our generator is eluted on Sunday night, so that we can do a synthesis on Monday morning. So it's very important. My experience is that anything less than 24 hours pre-elution is sufficient. If the generator sits longer being not eluted, then you're getting a bit of trouble with your synthesis.
Yeah. You have in-house built module for that?
It's an old syringe dispenser, to be honest. Yeah. It's automated by a clock, and it pops up at, I don't know, Sunday night at 6 p.m. and then the syringe driver eludes the generator and then it turns off again. So the syringe dispenser gets power, and that's all. So it's not a very complicated module we built. It's very, very easy, very efficient.
Yeah, that's a good solution. Any more questions? Yeah, please.
Can I just make a suggestion, Oliver, that you all stay here in Australia, because the regulatory environment in Australia is remarkably different, as you know, to Germany. We don't need an IMPD. All we have to do is fill in a CTN, which is a clinical trial notification, to our regulatory authority. It costs about $100. I think the price might have just gone up recently. And so it's much easier to do trials like this in Australia. And our exemptions for manufacturing, if you're in a public hospital, I guess, allow us to get around a lot of these things. Now, a lot of us choose to have GMP compliance, but you're not mandated to do that in Australia. So we've done a very similar trial to what you're proposing to do in really under 12 months, from the time that we got the money to the time that we finished the study. So Melbourne is a nice place. You should all consider staying here at the end of the conference and doing your research here in Australia.
I know it's very easy to conduct clinical trials here in Melbourne, particularly with Rod or Michael as coordinators. But the regulations are a bit not so tightened as in Germany or in Europe. And I think it's maybe a good idea to discuss in the tea break with all the participants from different countries how the regulations are and get more ideas.
One more question, Michael,
I gather when you started this process, you were going to perhaps use a variety of different gallium generators and you were validating them. And then when one got marketing authorisation, you settled on that one. But the next step is these synthesizer units. Do they have similar issues? None of them perhaps have marketing authorisation. Do they have labels about not for human use? And what will happen if one of these synthesizer units gets approval? Will you then have to restart from scratch and only use one synthesizer unit, or are they subject to different legislation?
No. I don't think that any synthesizers will get approval or marketing authorization. It's more like if you use different synthesizers, they will come with different kits and different methods, and that's what you need to harmonize. I don't think that there will ever be any synthesizer getting marketing authorization.
Yeah, may I elaborate on that, because I had also a question to you about that, because the manufacturing process, and you tried to actually harmonize even the chemicals that you used in the production, but I wonder if it's really necessary to do that harmonization, because what is most important is that you have formulated your product the same way and you have the same specifications and requirements to the quality of your product. And everything else should not matter, because as you mentioned also, it would cost a lot of money to do that.
Yes, absolutely.
And that would be just impossible.
That's absolutely right. I agree that fulfilling the product specifications should be sufficient, but that's depending on your national authority. If they require, you have to do the same synthesis process, and otherwise you can't do your clinical trial, then we need to follow these recommendations from the authorities. Or we can go into long discussions. That's the other option.
But I hope they would not claim that, because that would be...
They claimed it for our clinical trial.
Even conventional pharmaceuticals, they are not manufactured in the same way.
That's true. But I can only talk about our clinical trial and the requirements from the federal authorities. And I agree fully, if you have your product is on all centers the same, and you can achieve that, because you're doing quality control, you do your analytical procedures, and if your product fulfills the criteria, everything is fine.
Yeah, and usually those sites are GMP qualified and they're scrutinized by authorities, so they have the responsibility and license to do that. I have one more question. As you mentioned, there are several diastereomers when you have PSMA 11. And how do you justify to your authorities when you have at least two major signals that are seen in your HPLC chromatogram? as a quality control?
We published the data. It's
So
published, and
it's sufficient to just refer to Edwards' paper?
Yes, that's sufficient. They are familiar with that problem, and if it's scientifically proven that these two major peaks are PSMA in different diastereomers with same cell uptake and binding affinity, then it's okay for the authorities.
Yeah.
The authorities are sometimes a bit tricky to handle.
Well, thank you very much for your clinical trial, because you have paved the way for the rest of the world to do it in an easier way. So we have just to refer to your sites and your clinical
When it's finished, yes.
examination. Thank you so much.